Thanks, Evans. Thanks for the organizers for the opportunity to present our work. And thanks, Bram, for the really, it's nice to start with some passion in the morning, especially, after a world jet lag. So thanks for that very nice talk to open the session. And I'll tell you a little bit about the story of how we're trying to take this integrated approach. And we're trying to look at the host and the pathogen within the same project. We know a lot about the host, I think, but until very recently, in terms of the genomics of the pathogen, we had very limited information. 4 years ago, when we started, there wasn't even genome sequence of yellow rust. So we really had to come a long way to start understanding. We're not there yet, but I want to tell you some of the stories of where we're moving in that direction. And central to that has been the work that Diane Saunders has done as a new project leader. So she's generated in her lab really the tools to allow us to start looking at the pathogen like we couldn't before. And so we're going to get some very exciting data from her lab, I'm sure. And I'll show you a little bit about the work we're doing that she's been leading. And of course, we had to team up with pathologists as well, because we're trying to integrate the different layers of expertise. So we've also teamed up with Mogens Hovmoller, with Sarah Holdgate at NIAB, and with Subhash Bardwa in India as pathologists that can actually inform and help us in the genomics as well. From the host side, we have a series of other actors, including Brande, Ruth Wanyera, Worku in Ethiopia, and also Parveen in India. So the idea was to not just look at this problem from a UK perspective or an East Africa perspective, but try to actually include also India to really broaden how we're understanding this. Again, thinking about what we were talking yesterday and what Dave and what Marcel were saying about pathogens moving, we need to understand it not just from one country, but actually more as a global perspective. So I'll start off with telling you a little bit about the work we've been doing with the pathogen. So when we started off, there was very little information about the pathogen. Working with the pathologists that I mentioned, we've been able to collect isolates from from the countries that are shown here in red. We have sequenced about 40 to 50 different isolates at the genomic level. But of course, because yellow rust is quite a complicated beast, basically we have 2 different copies of every gene. It's quite difficult to assemble. So we've been working very hard to try to get a better reference of this genome. And what we have done, of course, is taken those isolates and done the pathology in terms of using differentials. And we've taken common differentials from Mogen's lab, bulk that seed, distributed, and we use the same differentials to do the pathology on these isolates. So we have, in a way, phenotypic data for those isolates. We have genomic data for those isolates. And now the idea is to try to put those 2 things together. And I'll show you an example. The other thing we had to start doing was that we didn't know the genes within the yellow rust. We had no idea. We could make predictions based on other species, but those predictions are often unsatisfactory. So what we did is that we took infected tissue, here highlighted in blue, and did RNA-seq, so sequenced all the transcriptome that was being expressed from that infected tissue. And we also took infected tissue from Haustoria. So isolated Haustoria to see what were the genes that were being expressed in that interface between the host and the pathogen. And by doing that, we're able to, first of all, generate gene models for yellow rust, but also generate, in the part that you see in red at the top, genes that were enriched within the Haustoria. So these are genes that are probably more interesting because they're right at that interface between the plant and the pathogen. So I'll show you a complicated slide. So basically what Diane was able to do was able to generate a pipeline that would take all this information, forget the details, but the important thing is that, again, taking that big data and trying to make sense of it is the really hard part. So basically using this and using several layers of information, we could actually distill that to a set of genes that we thought were of interest for the functional part. And this was published a couple of years ago. So based on that, we're able to generate a list of 14 putative secreted proteins that we thought could be of interest in this interaction. And here is where we hit the struggle sometimes, where we have to make new innovations because the technology is just not there for wheat. And in this case also, it exemplifies very nicely what international collaborations can do. So we're facing the problem of how do we actually express these proteins within the wheat plant. If you're working on Arabidopsis or Bentamiana and other systems, it's very easy. In wheat, it hasn't been done. But thankfully, when you have a group of international collaborators like here, you can actually email, in this case, Peter Dodds, who really, really kindly agreed to send us all the vectors, everything before publication, so we could start using it. And I think that's a real example of how this community is working and should work. So we were basically able to take some of this data and basically make us this graph, which basically shows in colors the different effectors, and we basically express those within the different differential lines. So those are the ones at the top. And where you see the highlight in yellow are basically the lines that gave us a response, a cell death response when we express these proteins in that particular genotype. So in this case, we see that we get a response with avocet wire5 and with Li, with wire7. So suggesting that we have a positive response when we express this protein within these two plants, but not in the isogenic avocet background. So that suggested something very interesting. And basically this is how the data looks. It's difficult to reproduce, but this is how it looks. So you can see the first two are our controls, which is basically the empty vector and effector 1. And then basically what you see here is the clone of AVR SR22 that Peter Dodds provided very kindly, as I mentioned. And then this is our effect. So when you see avocet, there's no effect. And when you actually express it in avocet with wire5, you see a very strong cell death response. So we now are making transgenics to try to characterize this effector further. We also started to understand the dynamics, not just at one time point where we did before of taking the RNA-seq and getting the expression. We also wanted to understand a little bit more about how those genes were expressed during the infection time course to learn more about how the pathogen and the plant were responding at the same time. And again, it's quite timely to do this because until very recently, we didn't even have a wheat genome. So actually, we can now have a much better wheat genome. We can take those reads and put them to wheat. We have some version of a yellow rust genome. We can take that and map it to rust. So we took different time points. We didn't sequence everything, just a subset of this. But basically what we see is that this is, again, analysis that's done by Diane's lab. And you can see that as the pathogen starts colonizing, there's very little pathogen RNA. But actually, by about 11 days, when you start getting perhaps the first pustules, you have roughly equivalent amounts of pathogen and host RNA. And that's quite important because if we did the same graph with DNA, the DNA would be very, very, very low. So if I want to sequence the pathogen at that point, it would be very difficult. But actually, if we can take samples from this point here, the 11 days, we actually have roughly the same amount. So that tells us we can enrich for the pathogen, we can see which genes are being expressed. And in discussions with Diane, with Sophie, and with other people in Norwich, we started thinking, well, can we actually use this as a way to genotype the pathogen? And this is something that Diane showed in her poster, but just to highlight this paper that we published earlier this year with this idea of field pathogenomics and now how this is now being worked with Dave and with others in terms of the surveillance. Because we can now take the leaf that's infected, we can crush it, we can basically take that, put it into a solution and ship it overseas without any problems. And basically with that, we can get information from the pathogen and from the host directly from that infection site. And importantly, this infection site many times is due to a single isolate. So we can actually get much information from that. And in this paper, there's lots more information there. So I'll tell you a little bit about what we're doing from the host side. And again, this has been a collaboration with several partners in Ethiopia, Kenya, India, and also in the UK. So what we did here is that this is using the Watkins population. So it's similar to what Urmil mentioned yesterday. But again, what she also mentioned was one of the problems we have is that these populations, once they were in a seed bank, they were distributed, but then it's very difficult to backtrack which is the seed of which, and the numbers get very wrong. Also, usually those gem banks have a collection of varieties or a collection of seed, not really a single seed descent. So here I want to recognize the work that Simon Griffiths did at JIC. So he actually took single seed descent for 3 generations to take individual plants from the Watkins, make them single seed descent to generate pure stock of that collection. That collection is now available through a seed store, so you can go online and order those collections. And this is part of a larger program in the UK, again, which is part of this wheat improvement strategic program that is looking at synthetics at NIAB, land races, and also ancestral gene introgression. So it's looking at a wide broad of germplasm to generate new resources for researchers in the future. And the idea was to generate the stocks that could be well archived and deposited in a place that anyone can access them without any IP. So they're completely free of any IP, MTA free, so people can use them. And you'll see why they're free of IP, because actually they come from many other countries of the people around this room. So they were collected, but they're really a repository for the world. And someone mentioned yesterday about getting more information, and you can find more information in this paper that, again, that Simon's group published on generating this curated version of the Watkins landrace cultivars. So we took the Watkins collection that was purified through Simon, single seed descent, and we evaluated in 2 locations in Ethiopia, also in Kenya, and we also at the same time evaluated this for stem rust and leaf rust. Although that was outside of our remit, we said it was a good opportunity to look at these populations like that. We also looked at them in England and also in India for yellow rust at adult plant and seedling. And what we wanted to do here was to, again, we talked with Urmil and we said, well, in this case, we wanted to look at some of the accessions that they hadn't touched upon, but also we really tried to focus on lines that gave us the potential for more durability. We had the first talk from Bob at the beginning about nomenclature and how important it is. You know, as a young scientist, you come up and you start reading early or seedling and adult plant and high temperature, low temperature, and all these kind of terminology, and it's quite difficult. So what we tried to think about was, can we find genes that actually give us some moderate level of resistance in the field across locations, and that at seedling do not have any major genes? That was the philosophy that we took when we were doing this. So here, these are the 23 accessions that we identified. So these are coming from all these different countries, as you can see here. There's a full list in the website, which I'll show you at the end. And these were, again, taking into account what Wolfgang Spielmeyer said a few years ago, that the need to generate some of these research in common genetic stock so we could compare across different continents what we were looking at. So what we said is that we took these accessions and we crossed it against Avocet S as the common stock, based on all the work that's been done here at PBI by Colin Wellings and others. So we took those, and basically we took 23 accessions that across the different locations had a partial resistance of less than 30 ms. So we're generating those as single-seed descents. They're right now at single-seed descent 4 or 5. Those populations, whenever they're ready, they will be available for anyone to study as well. We're going to be looking at them for yellow rust specifically, but they're also available for leaf rust and stem rust. So one of the things what happens is that once we generate those, how do we start doing the mappings? And we're trying to see how we can apply new genomic approaches to accelerate the way that we generate markers. And this is an example of how we're using bulk surrogate analysis, something developed by Richard Mitchelmore, who's probably in the audience, and how we combine it with RNA-seq to rapidly generate markers that would be amenable for breeding. In this case, what we did is we took a gene called WIRE15. This is a gene that's quite important. It's quite effective, I think, in all your countries. We are using it because it was very important in the UK, but the breeders were using, in that moment, microsatellite markers that were not really diagnostic in lots of the germplasm that they were using. So what we did is that we took the susceptible line and we crossed it to Avocet WIRE15. So we have basically, we generate an F2 population that's surrogating for WIRE15. And then when we have the F2, we basically screen that F2 and classify lines as susceptible or resistant based on their phenotype. And then we can actually group those into a group of plants that are susceptible, basically that lack WIRE15, and a group of plants that's resistant that has the WIRE15 gene. And we make 2 different bulks. And those bulks, we basically take them and just sequence the RNA of those plants directly. With that, basically, we generate lots of genomic information, but importantly, if there's a gene that's associated with resistance, it should be enriched in the resistant bulk. If there's a gene that's not associated, it should be basically, that's not associated, it should be found in equal frequency in the 2 bulks. So the way we do this is very simple. We take the parent and we first sequence the RNA. And this is not a hugely costly experiment to do because we can get the RNA, there's lots of it, and we don't need to find every SNP in the genome. We want to find enough SNPs to allow us to do mapping. So you can take the parents, find SNPs between the parents, and once we find these polymorphisms, we ask a very simple question. We sequence the susceptible bulk and we sequence the resistant bulk. And we ask the question, for every SNP, what is the frequency of that SNP in the susceptible bulk compared to the resistant bulk? So once we find the SNPs, we find SNPs that are actually coming from WIRE15, that are actually enriched here because they would give resistance. And if that SNP is really responsible or close to WIRE15, it should be absent from the susceptible bulk, because if it was present, it would give resistance. So very, very simple questions. So what we can now do is, because we have basically a very nice wheat genome, we can actually take all those SNPs and see where they go against the wheat genome. And you can see that we compare SNPs between the parents. There's lots and lots of SNPs between the parents. So that's great. We find lots of polymorphisms across all the genomes of wheat. But then if we ask the question, let's only take those SNPs that are overrepresented in the resistant bulk and underrepresented in the susceptible bulk, we basically come up with a very different story. We come up that almost all the SNPs that we find co-locate to chromosome 1b, short arm, right near the centromere. So basically the majority of the SNPs, which are highlighted here with color, with a bright color, all the SNPs actually coming from this, we simplified it to this. So very easy screens that can be done anywhere. By those F2, sequencing them, we get this information. But importantly, what we now have is we have an Excel file full of SNPs, but we don't have any markers for the breeders. So how do we do that? So one of the issues that we have now is this big data. We have too many SNP information. We have too many SNPs that actually, which one do we prioritize? So we came up with a very simple program called Polymarker. Again, taking advantage or the fact that the wheat genome has been released, one of the problems that we have is that when we try to move from microsatellite into SNP markers, a lot of times the SNP markers are not very good at separating homozygous and heterozygous classes because of the polyploidy. So we used a program called Polymarker, which is very simple, and the idea here is that when you design your primers, you take advantage of the information of the A, B, and D genome. And by taking advantage of the 3 genomes, you can actually make primers that have a higher chance of being specific to the genome that you're targeting. So basically, we're able to take those SNPs, use this program called Polymarker, and basically come up with lots of SNPs that look like this. So basically, you can separate homozygous classes very easily, and the heterozygous is somewhere in the middle. So now, in terms of doing marker system selection, you have a very clear assay where you have a heterozygous or a homozygous, and you can select that. Many times when people show this and they're not specific, you get the clusters that are very difficult to distinguish, and that makes breeding quite difficult. So we took those SNPs. This is a simplified map of that area, but basically we're able to find lots of SNPs around the region, and then 2 of the SNPs that were the most interesting ones were basically this haplotype, this R5 and R8, AT haplotype that basically was linked to susceptibility, and the GC haplotype that was linked to resistance. Because the GC haplotype was actually coming from the YR15 dicocoides introgression, it is quite diagnostic across lots of different germplasms. So this is basically a test of varieties that Limagran and RGT had from their diverse panel across the world, and it's basically diagnostic in all except one of their varieties. So it's a way to rapidly generate markers, tag regions, and although we didn't find the gene, it doesn't matter. We have 2 SNPs now that can actually tag the region, and breeders can now take that gene into their germplasm with much more confidence than before, where they were having to use many markers and microsatellites, which were quite difficult. The last thing I want to show you is how we're also using mutagenesis as an approach to help us in this manner. So we have a line called cadenza, which has YR7, and this is the typical YR7 resistance response. And working with Limagran again, with Simon Berry and Paul Fenwick, what we said is that we took cadenza and we mutagenized the whole population and organized it in a structured manner. And when we started screening, we found, of course, lines that were susceptible. So we basically have put mutations in YR7. That's great. But importantly, we also found lines that gave us enhanced disease resistance. So these are lines that gave us, within a family of segregating lines, we find lines that are YR7, and then we also find lines that have this enhanced resistance response. And we confirmed this in independent experiments. So we have now multiple mutants that are giving us this enhanced disease resistance response. But still, I mean, this has happened before, but one of the difficulties has been how do we get to those genes? So the nice thing about this is that these mutants are not just any mutants. These are part of a large collection that we've taken and we've actually sequenced their whole exome. So basically, we use this approach called exome capture, which Rohit mentioned yesterday, where you basically take the whole genome and you just capture the exons, roughly 1% of the genome, and you sequence it. So we've been able to sequence it and identify the mutations across all the protein coding sequence of these enhanced disease resistance mutants. The problem we have is that, of course, we have 5,000 mutations. Which mutation is the one that's underlying that phenotype? We still don't know. But the approach we're going to use is very, very similar to the bulk surrogate I showed you. So basically, we can take that YR7 cadenza line and cross it to the mutant. But interestingly, we're making a cross here of cadenza against cadenza. So basically, there's no background variation. It's cadenza against cadenza, but we know that this cadenza, the enhanced disease resistance, since we have sequenced it, we know it has 5,000 mutations. So now the question is very easy. The question is, when we screen the F2, right now we're at the growing the F2 population, so hopefully it'll be as easy as I think, but basically, when we grow the F2, the idea will be to do the same bulk, to score those lines with the same isolate, and find the enhanced resistance lines, and basically sequence them, and ask the question, from those 5,000 SNPs, which of those 5,000 SNPs is associated with that enhanced disease resistance? And again, even if we don't find the gene, we'll be able to get very closely linked markers and put this into breeding lines. So again, we started also the crossing of this cadenza mutant into several of the UK varieties, and then once we get the markers, we'll start doing the same against other germplasm as well. So with that, just to finish off by saying that as part of the program, I think we've heard it all many times during this meeting, the importance of having the next generation and the students. So one of the things that we really focused on is that sometimes in the UK it's quite frustrating because we cannot have international students on projects. It's really, really frustrating. You want to attract. I think whoever is giving the money here in Australia to fund international students has real vision, and I want to commend them because I studied with an international fellowship. A lot of people here studied with international fellowship, and unfortunately in the UK, we don't have that vision, at least for studentships. But thankfully, DFID was funding this program, so we were able to get international students in here. So basically, we have 4 students on the project, Mitali and Deepika, Mercy and Sisai, and also we collaborated with the project of René Prims and Leslie Boyd and Zach Pretorius, who were also working under the same scheme, and then Hester, which is a PhD student under their program, is also working very closely with Diane. And basically, we're using these studentships to look at different genes from Umbellata, Peregrina. Mercy has been doing great work. She just came back from Mogen's lab, where she's learned pathotyping, and with the new infrastructure that's being built at Calro and Jodo, we'll be able to actually do some of this pathology of yellow rust also on site, as we said was one of the things we wanted to do. And then with Sisai is also looking at Ethiopian durums. Again, he's made single-seed descent stocks of germplasm banks. He's taken 2 years to generate the germplasm, and finally now he's doing the genotyping and the phenotyping of that germplasm. But again, quite a lot of vision to generate the resource to now start doing this. And then Hester, you'll hear her talk quite shortly. One of the things that's also come from that is that Bram mentioned about hearing the farmers. In this case, we said we had in our plan to make a workshop for marker system selection and this and that. And the students ganged up and said, no, we don't want that. We have enough of that. We want to learn how to present a poster, how to give a talk, how to prepare a CV. So we actually generated a training on soft skills. So it's a 4-day training session on how to prepare a CV, how to interview. And we had our first session in Enjodo last year, and we'll have hopefully one in a couple of years in collaboration with Brandes Group in India. And the idea here was that just not apart from the training, but also the network that starts. And it's quite satisfactory to see that after a couple of years, basically, the 4 people here in this project are already starting to forge these relationships and discuss how to move forward with some of their ideas. So just to make sure that all the data I've shown here is publicly available, so the data from the Watkins Collection is in the Yellow Rust website, the collection of seeds is also through SeedStore, and then all the genomic data from the isolate is also available through this project website. So just to summarize, we've been looking at the problem of yellow rust from both the pathogen and the host. From the pathogen, we've done these complex genomic analysis to try to identify effector molecules that are important in the host-pathogen interaction. And also, by doing the RNA-seq, we've been able to develop a new way, again, of looking at the pathogen, and that's now helping with surveillance and things that we didn't initially set out to do, but actually have been very insightful. And then from the host side, we've taken these 23 Watkins accessions from the different countries highlighted in green. We have developed new ways to actually generate markers very fast, and importantly, to put those markers as SNP markers that breeders can use in marker system selection. And we've also been using mutant screens to find enhanced disease resistance mutants that hopefully now we'll be able to get markers very soon for and start breeding as well into diverse germplasm. So with that, I just want to acknowledge all the people working here. So I get the fortune of showing everyone's work, but it's really a huge effort by many people, especially Diane from the genomics side, and then people from Ethiopia, Kenya, Mogen's lab, India, and also from the John Innes and NIAB. And of course, thank you to the funders.